Enzyme activity is a fundamental concept in biochemistry that determines how quickly biological reactions occur. In this lesson, we'll explore the major factors that control enzyme activity and understand how temperature, pH, and concentration influence the rate at which enzymes catalyze reactions. These factors are crucial for maintaining proper cellular function and are essential knowledge for understanding metabolism and biochemical processes. Three primary factors affect enzyme activity: temperature, pH level, and concentration. Temperature influences the kinetic energy of molecules and enzyme structure. pH level affects the charge distribution and shape of the enzyme's active site. Concentration, which includes both substrate and enzyme concentration, determines how many molecular collisions can occur. Each of these factors can either enhance or inhibit enzyme function, making them critical control points in biological systems. Temperature has a dramatic effect on enzyme activity. Enzymes have an optimal temperature where their activity is maximized. At low temperatures, molecules move slowly, resulting in reduced kinetic energy and fewer collisions between enzymes and substrates. This slows down the reaction rate significantly. At high temperatures, excessive heat energy causes enzymes to denature, meaning the protein structure unfolds and loses its functional shape. Once denatured, enzymes can no longer bind substrates effectively, and activity is permanently lost. The relationship between temperature and enzyme activity follows a characteristic bell-shaped curve. As temperature increases from low levels, enzyme activity rises steadily. This continues until reaching the optimal temperature, where activity peaks. Beyond this optimal point, activity drops sharply as denaturation begins. For most human enzymes, the optimal temperature is around 37 degrees Celsius, which matches normal body temperature. This graph demonstrates why maintaining proper body temperature is so important for metabolic processes. Each enzyme functions best at a specific pH level, known as its optimal pH. The pH affects the ionization state of amino acids in the enzyme's active site and substrate binding region. When the environment becomes too acidic, excess hydrogen ions alter the charge distribution on the enzyme, disrupting the active site shape and reducing activity. Similarly, when conditions are too alkaline, the lack of hydrogen ions disrupts ionic bonds and hydrogen bonds that maintain enzyme structure. These changes prevent proper substrate binding and catalysis. Different enzymes have evolved to work at different pH levels depending on their cellular location. Pepsin, which digests proteins in the stomach, has an optimal pH of 2.0, perfectly suited for the highly acidic stomach environment. Trypsin, which works in the small intestine, functions best at pH 8.5, matching the alkaline conditions there. Catalase, found in blood cells, operates optimally at neutral pH 7.0. These examples show how enzymes are specifically adapted to their environmental conditions. Substrate concentration significantly impacts enzyme activity. At low substrate concentrations, the reaction rate increases linearly as substrate concentration increases. This occurs because more substrate molecules are available to bind with enzyme active sites, leading to more frequent enzyme-substrate collisions. However, at high substrate concentrations, a different pattern emerges. All enzyme active sites become saturated, meaning every enzyme molecule is constantly occupied. At this point, adding more substrate cannot increase the reaction rate, and the rate plateaus at its maximum velocity. This graph illustrates the relationship between substrate concentration and reaction rate. Initially, the curve rises steeply as increasing substrate leads to proportionally faster reactions. As substrate concentration continues to increase, the curve begins to level off, showing diminishing returns. Eventually, the curve reaches a horizontal plateau called Vmax, or maximum velocity. At Vmax, all enzyme molecules are working at full capacity, and the reaction rate cannot increase further regardless of how much additional substrate is added. This saturation phenomenon is a key characteristic of enzyme kinetics. Enzyme concentration has a direct relationship with reaction rate, but only under specific conditions. When substrate is present in excess, meaning there's more substrate than enzymes can process, increasing enzyme concentration directly increases the reaction rate. This makes intuitive sense. More enzyme molecules provide more active sites for catalysis. The relationship is linear, meaning doubling the enzyme concentration doubles the reaction rate. This principle is important in cellular regulation, as cells can increase reaction rates simply by producing more enzyme molecules. Let's summarize the three major factors affecting enzyme activity. Temperature follows a bell curve pattern, with activity increasing to an optimal peak before declining due to denaturation. pH also has enzyme-specific optima, with each enzyme adapted to function best in its particular cellular environment. Concentration affects follow saturation kinetics, where substrate concentration shows a hyperbolic relationship with reaction rate, while enzyme concentration shows a linear relationship when substrate is in excess. Understanding these factors is essential for comprehending how cells regulate metabolism and respond to environmental changes.
If you like this video, hit that like button and don't forget to subscribe. Visit codelucky.com for more such useful content.